Good morning, everyone. We'll start the webinar in just a few minutes. We'll give everybody a couple minutes to join. All right, we'll wait just another 30 seconds or so before we start. Okay, I'll go ahead and get started for us, and then as other people join, I can just jump in. So hello and thank you for joining us. I'm Tanya Marvin, the Marketing Coordinator at Simulations Plus, and I'll be handling the housekeeping for today's webinar. Our Director of Business Development, Dan O'Connor, will introduce our speaker for today. In this webinar, Professor from the University of Sao Paulo, Dr. Gabriel de Araujo, will present a recently published example from AAPS PharmSci Tech on the use of GastroPlus to assess the risk and pharmacokinetic relevance of different solubility and particle size between crystal forms of a poorly water-soluble antihypertensive drug for diverse psychological conditions. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. Dan O'Connor will be facilitating the Q&A today at the end of the presentation. So Dan, I'll turn it over to you now.
Thank you very much, Tanya, and welcome all. Good day to all of you. We have the great pleasure of introducing our speaker today from the University of Sao Paulo, which is in Brazil. He will be speaking on a very important subject about the pH dependency of the behavior of solubility as it impacts the pharmacokinetics of a drug. I had hoped that he'd be talking more about football and his nation's team. I'm sure it'll work its way into the conversation. Gold Cup and World Cup, all of that. But we're looking forward to taking advantage of his experience, which also includes, very uniquely, 10 years of industrial experience. So he has both the balance of industrial experience married to the academic approach. Currently, he's a professor of pharmaceutical technology at the University of Sao Paulo, and he'll be looking at what his research what it can do to accelerate the development of medicines and improve their safety and efficacy as well. But before I turn it over to you, Gabrielle, I'll throw it back to Tanya and invite the first poll question.
Okay, let me just launch this poll and we'll give everybody a couple seconds to respond. So how long have you been using GastroPlus? So please select one. Got the results coming in. We'll give it another couple seconds. Still have a few results coming in. Okay, it looks like we have everybody. Let me just share those results. So most of our users are under 1 year.
Okay. So that's great news. Okay, well then we'll all learn together. Without further ado, let me introduce Professor Araujo.
Thanks so much, Dan and Tanya, for the introduction and for the kind words, and not to mention the football part. Okay. And I also do thank Simulations Plus for the invitation. And for this opportunity to show this work that we conducted at the University of São Paulo. And hello everyone, and thank you for being here with us. It's a great pleasure to be here with you. And I hope this webinar can contribute with your work, or at least to gain some insights on the issue. Okay. To start, first of all, to start this presentation, I would like just to do a brief introduction about the University of São Paulo. We are here in South America, São Paulo State in Brazil. We are the largest university in Brazil, and our School of Pharmaceutical Sciences is a very traditional school in Brazil. And I would like to take this opportunity to thank also Simulations Plus for supporting us with academic license, and all the team for that always help us during this year, John, the bag, and all the team. Since 2015, we have been teaching undergrad students, over 500 students, concepts of biopharmaceutics and drug delivery with GastroPlus. As you can see here, they are very happy with that. Of course, before test, but it's really, they really appreciate that. It's really great for their careers.
Let me interrupt for a moment and ask, are you sharing the screen? Because we see your face, but I don't see the screen.
No,
Tanya?
okay. I'll try again.
There we are. Thank you.
Perfect.
Okay, I'll try again. Is it working?
Yes.
You had it just a moment ago. There we go. Perfect. We see it.
There we go. Okay. Okay, this is the HAP student. Okay, we are really thankful to Simulations for the support with the academic license. And so here a little bit of our research. Okay, we work with this interface between solid state chemistry and pharmaceutical technology. So we work with the development of amorphous systems and the study of drug polymer, drug excipients interaction in amorphous solid dispersions using high energy X-ray from synchrotron source. Also develop drug delivery systems, and we are having a lot of fun studying with acoustic levitation for the development of new amorphous systems, and also for accelerated screening of formulations. Also, we are working on the polymorphism of drugs to help characterize poor characterized drugs and help the quality control. So for those who are not familiar with polymorphism or cannot remember, let's just a brief review. Polymorphism is this phenomena where chemical entity has the ability to crystallize in different phase with different internal structure, let's say, but different unit cells and different packings, okay, and that sometimes different conformations in solid state, and you can easily detect using X-ray powder diffraction analysis. So this change in internal structure modifies important physical chemical properties. Here we are going to focus on the solubility and dissolution because of the impact on the bioavailability, but it can change hygroscopicity, stability, crystallization, also have great impact on analytical methodology. For example, vibrational spectra can change and it can cause impact on quality control, and some very important physical chemical properties like melting. So for example, here, for example, in the food industry, change of the melting point of cocoa butter that can really impact the quality of chocolate. This is a great concern for those who are chocolate lovers for, as I am. And so the occurrence of polymorphism is really high. It's estimated, depending on the statistic, for sure over 30% of the APIs shows polymorphism. So probably you have heard about cases like ritonavir and others with great impact on bioavailability. And one very important point is this relationship between the stability and solubility. Usually the most stable polymorph is has the lower solubility, okay, and the unstable form or metastable form, which has higher free energy, has higher solubility. So the problem is that they tend to convert to the most stable form during stability. When we evaluate the solubility advantage of different systems, when we evaluate, for example, highly disordered systems like amorphous, this increase in solubility goes higher than 1,000-fold. However, when we are dealing with polymorphs, this relationship between the most soluble and the lower soluble form, the lowest stable form, is about 2, because the difference in lattice energy is very low, like 5 to 6 kilojoule per mole. So, however, there are a lot of reports with ranges from 20 or 30, and this usually is the drugs that came to us in a very important project. So this is really can be a really great problem. Well, one point that we see in literature is that most of the studies on polymorphism came from from chemical engineering groups or groups that has great background in chemistry. They do a great job in thermodynamics and structure, but sometimes the bioavailability relevance are not addressed. And when we see the conditions that the solubility is evaluated, most are in pool water. Sometimes have mixture between organic solvents and water. Sometimes intrinsic solution relationships, or different apparatus, some surfactants. But this relationship at the maximum 2 came from this kind of data. So if we see one information that the solubility is very important for this polymorph, does not have a direct relationship with the bioavailability. You cannot assume that, okay? Another point that we have to evaluate is the possibility of phase transitions in solution. So we start with one polymorph and then it transforms to another form, to the most stable form, for example. Okay, here an example of the erlotinib tyrosine kinase inhibitor, where we have here in red the less stable form, the more soluble, and in green here the form with lower stability and lower solubility. So when we have a batch with mixture between them, we accelerate this transformation, and we can see the less stable form changing, precipitating into the lower stable form. Okay, so this is very important. You evaluate this possibility of phase transition, solid, solute, solid transitions. Okay. So here today we are going to show you the study of polymorphism of Lercanidipine hydrochloride. This is Elias. He was doing his master's research on the solid state of Lercanidipine polymorphs. due lack of information. This drug, it's a antihypertensive drug. It's a poor soluble, very low oral bioavailability, very high log P. So he was studying, trying to synthesize, to crystallize a single crystal, studying isolated forms, studying the relationship between different solvents, trying to find out if the stability relationship is monotropic and anisotropic. And we found very nice results about photostability. We found that form 1, the metastable form, also has lower stability regarding to photo degradation. So the photostability is really lower than the stable form. And he was very happy in this solid state world. Okay. However, we also were studying the possibility of phase transitions in different solvents, including water. Okay, to see if is there the possibility of the one form transforming to another form in any solvent. So we found also some inconsistent reports saying in some patent documents that saying that Form 1 exhibits poor bioavailability in dogs and humans than Form 2. And we thought, oh, this is not right. Our data shows that Form 1 is really the higher solubility form, and it's not. There is something wrong in that. And also, there are some data in regulatory documents that shows some specification to Form 1. So there is a concern of the amount of Form 1 in raw material. Okay. Then I asked Elias, are you about to defend? So, but I think it's important we explore a little bit more experiments. Have you finished writing your dissertation? No, not yet. Okay, let's do a little more, a few more experiments. Then just to see this relationship between the solubility Form 1, Form 2 in pharmacopoeia buffers. Okay, then you can explore the pH range and have this data to complement the study. He said, okay, let's do it. Then what we were expecting, of course, this is the main species. This is a weak base pKa 6.8. We are expecting a higher solubility at low pHs. And then we found something really not usual. The relationship between the solubility of the forms changed depending on the pH. Okay, so, and this was not expected. And also we saw that at pH 1.2 and 2, very low pHs, the solubility was really, really low. So this is it's a great chance to teach the students another very important law in science, the Murphy's law. Okay, so say, Elias, don't worry, everything will be fine. We can, of course, cannot sleep with this information, need to explore a little bit more, but why not try to explore this information with others in other ways, okay? So here we can compare the buffers, the ionic strength of pH 2, for example, in almost in the same level of ionic strength, almost very similar here. The impact on solubility is really high. So we realize that, of course, we are dealing with a salt, a hydrochloride salt, and we have the common ion effect. In this case, this effect is really pronounced. Okay, and depending on the species, the ionic species in solution, we have a very different influence. Okay, so since that in phosphate buffers, we can really see this difference between the solubility of the forms. So it's very important we consider that. Also, take note, this is a very good procedure to do when we are studying different polymorphic forms, or to collect the precipitate, okay? And you analyze it using X-ray. So we did that at the end of solubility study. We did that for all samples, and we really checked that there were no phase transition, okay? We are really studying Form 1 and Form 2, and they did not change phase during the solubility study, okay? No other form or salt or something was being formed, precipitating during the solubility study. This is a really good practice. Then we carried out a few more experiments, of course, trying to very diluted buffers. So, and then with diluted buffers, we could see this difference increasing, okay, more pronounced. We tried also some intrinsic dissolution. We couldn't see any difference, probably due to the pressure applied, and also using kinetic solubility studies at pH 1.2, we could see that even with low solubility levels, you can see this difference between Form 1 and Form 2 very pronounced. Okay, so after all this data, some questions come up. If I have tablet with Form 1 and a tablet with Form 2, are they interchangeable? Can I have, for example, if I change manufacturer, or I know that there are changes in manufacturing holds, there are batch-to-batch variability, and why not some commercial opportunities? Okay, that sometimes we need to change the raw material, sometimes do patent issues. So if I have one tablet with one form and another tablet with another form, are they can they be made bioequivalent? And also knowing that influence of pH, what are the effect of physiological conditions such as changes in pH, stomach pH, in the bioequivalence of those forms, okay? And also, is there any way to set specifications in particle size to counterbalance this difference in solubility? Can I modulate that with particle size control? Okay, so and of course, we're talking about a generic drug, and to conduct new clinical studies, PK clinical studies, it will have very high costs. Also, to sacrifice lives in animal models will not be I think to get just basic information, I think is not just does not justify. So it's really nowadays you can really count with in silico simulation like GastroPlus to gain information, and if necessary, goes to in vivo studies with more certain of what we are going to obtain and decrease the number of animals, etc. So to get a lot of information to access the risk can be done with GastroPlus. So we decide to evaluate this, what's the relevance of these solubility changes using in silico approach, okay? So Tonya has a poll now. I would please, Tonya, take it away.
All right, thank you, Gabriel. We have one more poll for everybody. Launch it here. So what other events do you intend to participate in this year? Please mark all that apply. Got some results coming in. A few more seconds. A lot of them coming in. All right, it looks like we have everybody's answers. A few more coming in. All right, I'm going to go ahead and close the poll. Let me just share these results. Wonderful, GastroPlus webinar. So, Gabe, we might need to have you back.
Okay.
Thank you.
Is my presentation in the presentation mode?
It is, no, it's in edit mode.
No.
Yeah, it seems like we're back to the original slide, number 1. Oh, have we lost him, Tanya?
Let's see. Are you still on with us?
Okay, what about now? Is it working?
You're back now, yes. You're on. You're showing slide 1.
Oh,
So
that's weird because I'm in slide 20. Okay,
all right.
I will stop just right now.
Every platform has its idiosyncrasies. What are you seeing, Tanya?
I see
Okay.
slide 1, yes.
Okay, let's try again.
And I see it, slide 1, it's in edit mode. There we go. Perfect. Now you're on slide 20.
Okay. I think you got used with this during pandemics, right? Okay, thank you guys for your patience. To answer this question, first question, of course we need a model, okay, and we have literature data with PK clinical data from tablets. However, when we are dealing with solid forms, we have you know that this is a BCS class 2 dissolution limited absorption. So we know that there are a really great influence of the particle size. So then we decide not to, and you don't know which particle size the drug, the tablets used in the clinical trial had. So we decided to look for PK clinical data of drug in solution. Then we could only skip this solution phase and work and optimize other aspects of the absorption phase. Okay. So our strategy was we construct the model based on the PK, the clinical PK study of our oral solution, which was administered in soft gelatin capsules. And after that, we have a model. We used the built-in IR tablet, loaded the PSD and density of data of the formula, and then we compared the results with the clinical data from literature. So once we have a good model, we can run the IR tablet with form 2, run parameter sensitivity analysis, and evaluate the change in gastric pH. Okay. So we obtain a really reasonable model with the oral solution here, as you can see. Some remarks here. We had to, at first we started with some data from KQ2 cells, but we realized that it was not, we are having a lot of trouble to adjust the model. And then we realized that KQ2 has some limitation capacity to model and to predict in vivo absorption of molecules over 600 Daltons. So we could optimize this permeability and achieve a really good model. And also there is a really impact of enteropathic circulation. So this was really important to this model. Then with this model, we loaded real data from particle size, from experimental particle size distribution for form 1 and form 2, also some density data from aeropicnometry. And this was one of the most nice parts when we just change from solution to IR tablet using the building, and it matches perfect. We had a really visible model matching with another study from literature with tablets. So and the results showed us that both forms in fasted state and are bioequivalent when administered in tablets. However, we couldn't sleep with the information that at pH 3 we have the difference, the solubility difference between the forms are higher, and also this is a dissolution limited drug, right? So we want to know the particle size influence. So GastroPlus allowed us to simulate, compare the particle size distribution, and we've increased a little bit the particle size, then the impact on in vivo dissolution, and also comparing fed state and fast state. And we saw that the only condition that really matters is at pH 3 when the stomach in fast state at pH 3. So what we see that we can really counterbalance the influence of polymorphism on solubility. Okay, but form 2 is very susceptible to this to particle size at this pH. So also we tried to evaluate this relationship between Cmax of form 2 and form 1 and in the AUC form 2 and form 1, and to see if they are out of the limit of bioequivalence. So once again, no influence of particle size on fasted and fed state. However, when we change to stomach pH 3, since that when we are have higher particle size distribution over 20 microns, this can be really a really risk for the bioavailability. Okay, and we know that form 2 is really more susceptible than form 2 is more susceptible than form 1, and regarding the particle size. So, and these low gastric acid conditions, nowadays we live in a really crazy world with everybody, a lot of stress, and a lot of people taking medicines like proton pump inhibitors like lansoprazole, omeprazole, that can change the stomach conditions in fasted state. Also, some populations have higher stomach pH. So this can really a concern for some cases. Okay. This is a very nice tool that some recent versions of GastroPlus have. You can input the solubility and proportions of each form. Here going more in the compound tab, more solubility, and you can add the solubility, and the proportion of the forms can be not only polymorphs, can be amorphous, and etc. This I didn't have the chance to test this in this study specifically, but I really want to do this, and I really recommend you to test, and I'll be glad if you share your results with us, because since a really interesting tool to be used in the polymorphism studies. Okay. So let's some conclusions. Form 1 is really the most soluble in form in aqueous medium, so there is no doubt. However, this relationship between the solubilities varies according to the pH. Okay, it can be from 0.9 to 3.9, depending on the buffer nature. Also, we have higher pH at low, higher solubility at low pH. However, we have to be careful with the common ion effect. Okay, this is very important when you are dealing with this salt. And PBPK simulations showed us that both forms with proper if you set a specification of particle size, they can be essentially bioequivalent under normal conditions. Okay, but it's very important when you are dealing with ionizable drugs, ionizable polymorphic substance, you consider the influence of buffer nature. Okay, this is really very important. And also a very nice tip for those who are doing PhD, finish your thesis. This is before your advisor give you more suggestions of experiments. This is Elias. He did a really hard work to complete all the experiments, and we are really happy that this work was achieved. And there are other works coming on from this, the structural part, which was the main focus of the work. But this was really nice, the tour of the work. So if you want more information, more details, there are a lot of supplementary material in your publication. This manuscript was published in the AAPS PharmSci Tech, and there are details of the parameters used in doing simulations and also other information in supplementary materials. And I'd like to thank all the collaborators who worked really hard to make it possible, and also our support of Simulation Plus with academic license and the funding agents for this work. This is my contact. Please reach me out if you have any questions. And thank you for your patience and your attention. We are open for questions. We'll try to do my best to answer that. Okay, thank you very much.
Well, thank you very much. This is Dan again. And we do have several questions, but also I'd just like to extend two invitations. First, as Gabrielle mentioned, for anyone that might be interested in learning more about what we call the Simulations Plus University Plus program, please do visit our website. The Simulations Plus University program provides access for qualified universities and programs to have free access to any of our licensed platforms. So for your research, it could be invaluable. And then the second invitation is to invite a few more questions if you like. We do have a couple of questions. Let me see if I can pronounce them correctly. First question is, are phase transitions in solution affected by the solvent? For example, does the composition of bioavailable media affect this equilibrium? Gabrielle?
Yes, they can, because they can change the solubility, right? So if you change the solubility, you know, when we have electrolytes in solution, we have decreased the solvating capacity of the medium, right? And this can and then this, you know, the salting out effect. So this change solubility, then change the kinetics of phase transition.
Okay, great, thank you.
Did I answer the question?
Well, I don't know. That would be for you to know. I don't know, but I think so, yes.
Okay.
Next question, isn't the CACO2 data more likely to be compromised due to low recovery or cellular binding in the permeability assay due to the high log P of LHC?
Yeah, that's a good question. We assume that this is really great question, but we assume that this we assume that the main problem was the permeability, because we tried a lot of different ways with transporters, etc. And this seems that the data was not the only thing that during optimization seems that CACO2 was really the permeability data was really the great concern. As we know that This is a class 2 drug with high lipophilicity, and you know that's the limitation of the KQ2 to when we have high molecular weight drugs. So we decided to go with this to optimize the permeability. But of course, there is this point regarding to the recovery and cellular binding, and this is a really lipophilic drug. Also, you know that most of the transport is transcellular transport. So yes, this can be really a great point.
Thanks very much. Do you have any information on the intermolecular interactions in the two crystal forms?
We are struggling for that. We crystallized we obtained the single crystal of the form 1, and now we are going to publish it. And some of the other form we couldn't achieve the single crystal, so we were trying to solve the structure using powder data, which is really hard to do. So for one form, yes, we do, and we're going to publish this year for sure. And the other one we couldn't obtain until now, but we are working really hard to get it. Yeah.
Okay, great. Thank you. Questions continue to come in. Thank you very much, all. How did you consider the HCl salt disproportionating in vivo in your GastroPlus model? Is that necessary?
Okay, good question. We used the building PK-based solubility model that GastroPlus has by fitting it to our experimental data. Okay, we used our experimental data and fit with the building model that GastroPlus had.
Okay, great, thank you. Form 1 and Form 2, the major excuse me, losing my coffee. Form 1 and Form 2, the major energy barrier can be balanced by pH. Is that correct?
That's a good question. That's a good work to be done. If I suggest that to Elias at that time, he not sure if I'll be here today. But that's a really great question about the energy states and the energy barrier to phase transitions using different pHs. That's a great question, but we really don't know that. This is a really great question to be explored. One information that's not related to that, but kind of, is that we had to use the big salt effect on the GastroPlus too. So this was really important to our model.
That's great. And science is always about the next discovery, right? So there's always
right. And I would take note of this question because this can be really a good question to be answered.
All right, we have one more question here. When analyzing particle size variability, do you assume a uniform distribution for each simulation? For example, all particles are assumed to be the same size.
No. We used about 10 beans to do that, to cover the particle size distribution. We tried to match, do the best to match our real particle size distribution.
So is it safe to assume then that a non-uniform distribution would not impact your results, or would it?
Yeah, the distribution was not the same. It was uniform, a non-uniform distribution, right? It's a monomodal unit. It's a monomodal distribution. So we had different particles. We didn't use that. There is an option to, I think there is an option to consider all the same size, right? We're using medium, but we prefer to divide in beans and try to cover it.
Right, okay. We have a little bit more time. If there are other questions, I'm on the last that's been asked. It's a question probably for me, which is how do we sign up for an evaluation of GastroPlus that's very simple. Just ask. We have a website where you can request an evaluation of GastroPlus, or reach out to Tanya or me, Dan O'Connor, and we'd be more than happy to provide that reference site or evaluation license to you. We do have another question. And again, we have a few more minutes left in the hour. When you were simulating the oral solution in GastroPlus, what is the rule of assigned reference solubility? Can the reference solubility be increased using a solvent like labarosol?
Well, what we did for the oral, we loaded our experimental data and used the built-in PK-based solubility model that GastroPlus have, and used the fit to adjust the solubility profile and PK profile. Okay, so it's a mix between our experimental and the GastroPlus built-in model. So if you have this, if you have chance to have our solubility in laboratory, probably you can try to input that and try to adjust the model based on your change of solubility.
All right, thank you. Another question just popped in. Did you need to use IV data to build your model as well?
Of course. If you IV data, I assume that's in vivo data, right?
I don't know if it's in vivo or IV.
Okay.
Yeah, I don't know.
IV intravenous probably. Oh no, yeah. I will try to answer all these possibilities. Okay.
Yeah,
so
okay,
okay. First, if it's intravenous data, we didn't. I just worked on the oral solution model data, PK's oral solution. Regarding, of course, all these assumptions and all this risk evaluation that we did needs to be validated with in vivo models, okay? And there is as any simulation. However, for some simulation, due ethical reasons, I don't think for some cases like this, for ethical reasons, I don't see that it's worth to conduct clinical studies if the gain is not, and if the gain, the cost is not justify it, okay? If you want only exploratory information. So okay, that's, I'm not sure if this answer this question, but
yeah, I think that does. And
you can send after. I can try to advise better.
I mean, that's one of the intrinsic benefits of modeling, is that you don't expose to unethical or harmful research. So that's one of the fundamentals, if you will. What about dose-dependent exposure? Is it linear?
Yeah, it is linear. It is linear.
So there were no issues with that?
No, it's linear.
Okay. All right. Well, it concludes the question and answer. Thank you very much for the questions. And most importantly, thank you very much for the answers. And without further ado, let me turn it over to my colleague, Tanya, for a conclusion.
All right. Well, thank you, Dan. And thank you, Gabriel. That was a very informative presentation. And as Dan mentioned, we do invite everybody to learn more about GastroPlus and our University Plus program on our website. It's www.simulations-plus.com. And I did put a link in the chat to our University Plus program if anybody's interested in learning more about those academic licenses. The webinar has been recorded for playback and will be available on our website and our YouTube channel. And that concludes our webinar for today. It looks like we got to all the questions and answers. So thank you for attending.
Yeah, and we did learn that Brazil is qualified for the World Cup this year. So congratulations.
Let's talk about other things.
Have a great day. Thank you.
Thank you, Dan.
Thank you, everyone.
Thank you all for attending.
Thank you, Gabriel.
Bye.
Bye-bye.
Take care.